Hello, everyone. Thank you for joining us on this episode of BAEP Talks With. This is a discussion with a person of interest who lives with, manages, treats, and/or supports others with a chronic menstrual health condition in whatever capacity that support may look like. My name is Julia Mandeville. I am the co-founder of the Barbados Association of Endometriosis and P.C.O.S, and I am your host today. And today I'm really excited because we are joined by one of my former lecturers, Dr. Angela T. Alleyne, PhD, MPhil in biology, to discuss her research focus in uterine fibroids in Barbados. So we have a PowerPoint here, which is a first for BAEP Talks With. So again, another exciting part of this presentation today. So permit me a little bit to share some more about Dr. Alleyne. She gained her PhD from the University of the West Indies Cave Hill campus in 2000 and was a postdoctoral research associate at the University of Nebraska, Lincoln in the plant pathology department from 2001 to 2003 and an assistant professor biology at the Edward Waters College in Jacksonville, Florida from 2003 to 2008. She joined the UWI Department of Biological and Chemical Sciences as a lecturer in biochemistry in 2008, and in 2017 she was awarded the UWI Principal's Award for Excellence in Teaching, Research, and Service. Dr. Alleyne has written over 30 peer-reviewed articles. She also serves as the program coordinator of the Master of Science program in Biosafety at the UWI. Her research career has focused on the use of molecular biology techniques in understanding and diagnosing both plant and human diseases, specifically uterine fibroids. The aim of her work in uterine fibroids is to examine the allelic distribution of gene exhibiting genetic mutations caused by the substitutions in the genetic code. Hopefully we can break that down during the discussion for everyone to understand. In a mainly Black population that has not been previously widely studied. Welcome, Dr. Alleyne. Thank you for joining us today.
Thank you, Julia, for that wonderful introduction. And so, yes, I know it's a mouthful, the work. And so today I'm going to focus mostly on, well, only on the work that I've done on uterine fibroids. I know it's a strange mix to mix plant pathology and human diseases, but the techniques we use in both of these endeavors are basically the same techniques. And the genes we study have a similar manner of expression in exhibiting diseases.
So could you tell us a little about your professional journey?
And so I will, okay, so next. Okay, and so it's interesting that you've asked that question. It's a bit of a point of reflection for me to answer that question, and I don't normally take the time to reflect on something like this. So I'm glad that you asked that question. And like you said, my journey began with a bachelor's degree in biochemistry at the University of the West Indies, UWI, as we call it, in Mona, Jamaica. For those of you who don't know, although I live and work in Barbados, and I did my first degree in Jamaica, I am from another island in Trinidad and Tobago. And so when I first came to Barbados, many people thought that I was from Jamaica, but I did my first degree in Jamaica, and then I went back home. And with any story about an academic career, there are stops and starts, possibly. There are diversions along the way. So the straight story is, as Julia said, it was a BSc at Mona, an MPhil and PhD at Cave Hill in 2000. Then I came back to Cave Hill, where I lecture biochemistry and so on. I found my way to Cave Hill serendipitously, probably because I got a call having done molecular biology research, courses at Mona. They were looking for someone with molecular biology, and I was able to get that job there as a research assistant at Cave Hill. And from Cave Hill, from that job at Cave Hill as a research assistant, I continued doing work from my PhD and MPhil. And the work there was heavily into molecular biology and biochemistry, although you were studying a plant disease. From there, I did go to do research at the University of Nebraska in Lincoln, Nebraska, again in the plant pathology department. But after 3 years of a postdoc, I then moved on to a small HBCU those of you who are not in the US, historically Black college at in Jacksonville, Florida. And I was an assistant professor there. And so there was no opportunity to do plant pathology research. And it was at that point that I began to do a lot of work in molecular diagnostics, bioinformatics, metagenomics, and continued upgrading my skills and did 2 postgraduate certificates in clinical molecular diagnostics with the aim of coming back to Barbados and starting my own diagnostic lab. Fortunately, actually, when I was on my way home with this enterprise in the back of my head, I got the call for this position at the UWI Cable campus, and I took up the position as a lecturer and now a senior lecturer at UWI Cable. However, I still engaged in molecular diagnostics research and used the techniques that I developed along the way to enhance my research. And it was at EWC actually, and at that stage in my career that I got an interest in fibroids.
Okay, thank you.
I hope that journey is winding, but it explains how we got here.
I guess I would like to ask, what specifically led to your research interest in uterine fibroids? With all of the, the expertise that you have and the knowledge that you have in molecular biology, why uterine fibroids?
So I've just explained the variation here again. When we talk about variation, we talk about people, and I specifically look at populations. So I'm going to get back to that in a minute, and that will answer your question.
Mm-hmm.
When we look at populations, we tend to think, we know, having sequenced the human genome, that we're about 99% similar, where we could be in terms of our genes. Mm-hmm. We are all of the same species, and therefore when we look at the genes, we're 99% similar. But that 1% difference, it makes a big difference because it reflects the expression of genes in terms of the importance of the environment and how these genes work. Mm-hmm. And so when you think about variation in populations, it's about the expression. So I got interested in fibroids simply because when I was doing a routine checkup one day in Jacksonville, it's a simple medical, and I think this is true for most women, I was told I had fibroids. It was a gynecological check. And the doctor didn't think there was anything make a big deal about it. But I asked more questions, and she was saying, "Oh well, most women have these, and in fact, Black women have a lot of it." Mm-hmm. So I thought I have never had any illnesses in my life before, and I asked my sisters. I have 5 sisters, and so I asked, and 4 of them had fibroids. My mother had fibroids, I was told. My grandmother. And so I was like, if so many women have had this condition, why don't we talk about it? Why don't we tell young girls about it? Mm-hmm. And that made me think something needs to be done. And so I am quite happy to see you, Julia, and your colleagues, former students of mine, in this realm of advocacy for condition, because I think young women, young Black women need to be made aware and need to be talking more about it. And I'm not speaking about, when I say Black women, actually, I'm not even talking about in the United States. I'm actually speaking about in the diaspora, in the Caribbean community, in African societies, where I think these conversations need to be had because it affects us greatly.
I completely agree with you there. And so thank you for the research that you're doing, because whilst the advocacy is really critical, obviously we need to back that advocacy up now with the facts and the figures that come from the research that goes on, behind the scenes to help us, share with relevant stakeholders. This is the impact of these conditions. This is the proof that these conditions do impact our population. What can we do about it now to make life a little easier for those who would be diagnosed with it? So thank you.
You're welcome. Yes. And so that was my response. As a scientist, having done some work, as I said, in clinical molecular diagnostics, and with the idea of going back to the Caribbean, to Barbados, to do molecular diagnostics, I felt one of the first things I would want to do would be to engage more with this condition that affects Black women, because it just made sense. It made, you know, and having read the literature, I saw that while there were studies being done in the United States, in Europe, and even in Asia, when you look at the cohorts, Black women or women of Afro-Caribbean descent were not highly represented. I must admit that that is changing for fibroids, but at the time when I started this research in about 2010, 2008, 2009, that was not the case. And so, yes.
So outside of, I guess, raising the level of, raising this to like I guess, a public health and a critical research level, what else do you want to achieve with this particular research?
Well, I really want to basically have us represented in the data. And I say this because when I read more about in the literature, I see evidence of Black women, but not Afro-Caribbean women in the data. And even though Black women are said in the literature to have this condition more, we're not a monolith. Our experiences are different, and also the expression of the genes would be different in our environment. And so I would like to see more of the work looking at African countries as well as Afro-Caribbean women, islands, if you want to call it that, within the data that is presented as Black women.
Right, because as you said, we're not a monolith. How does your research and the results of your research compare with others in the area? Is there a lot of consistency in the area?
So, like I said, our research on fibroids was looking at the condition and the differences we find in our population. And I'm here speaking mostly about the Caribbean, specifically a Barbadian population. It's interesting that I live and work in Barbados, because of the Caribbean islands, the Barbadian population was fairly unique in the sense that it had a fairly homogeneously Black history compared to other countries like Trinidad, Jamaica, Guyana, where there's more racial mixing. So I was fortunate to be in Barbados and sample this population. And interestingly enough, in the databases, that is the genetic databases, Barbados is represented more than some of the other islands in the region. So when we began looking at this, the whole point was to think about estrogen. That's where I began, because thinking about it as a female condition, having read the literature, it all said that it had to do with hormones. And several things were put forward, because, for example, we know that, or it is said that, post-menopause, the condition is ameliorated. I wouldn't say it goes away, it's ameliorated. For Black women, it is still there. In young girls, in Black females, it starts earlier. Sometimes you see that they menstruate earlier, and so they tend to have more of the complaints associated with their menstrual cycle than other groups. And so all of these things we know or we thought about, but the simplest thing was to look at the hormones. And so when I began this work, I thought, let us look at genes that are coding for estrogen. This is a very complex diagram, so I'm not going to spend too much time on it. But we have here one of the genes we looked at something called CYP17, and another one called CYP1B1, and this one called CCDC57, which is associated with fatty acid metabolism. Now, the early work that I had read suggested that this particular one, CCDC57, was associated with white women. This was in 2012. This study came out around 2012. And it was new at that point to think of a single gene, being associated with fibroids. Prior to that, there had been work done on chromosomal breakages, for example, being associated with fibroids, some particular chromosomes. I think it was chromosome 11. I'm not too familiar with that right now, but chromosome 11, and I think it was 17, something like that. Was breakages. When we say breakages, I mean faults in those chromosomes were associated with fibroids. But a single gene until 2012 had not been looked at.
Right.
But the research indicated that this particular one was associated with fibroids, but mostly the data set only had white women in it. Subsequently, there was a study done in Brazil linking estrogen in this particular gene, an early gene, CYP17, and they felt, or they saw that there was a link between this gene and a change in that gene and uterine fibroids in a sample of Brazilian women. Now, if you think about Brazil, there are Black women in Brazil. They are not, there are mixtures and so on. So that led me to want to investigate this particular gene. And then my readings also indicated that in Asia population, the CYP1B1, again, a change in that gene was significantly associated with fibroids. Right. So first we began looking at this one with undergraduate students in 2010, around there. And we compared what we found with so this is the work we did with the CYP17. A single gene gives us one variant, a change in that allele gives us the other variant. And so we published that work in 2014, and the main finding was that there was no significant difference in terms of the population in Barbados we sampled and that change in that gene or variant. Unlike the Brazilian study, that one study that found that there was an association. However, we also measured things like the BMI. We measured estrogen levels a year later, and found, as other studies had found, that there is a significant association with weight or your waist circumference, for example, and also levels of estrogen. Right. So for us, it established that estrogen levels are important. Estrogen is important as a hormone in how we regulate our metabolism as women. Even not only estrogen, but progesterone. These two things, our male-female dynamic is so, it's such a fine line. And so estrogen helps us, for example, increases or decreases our risk of heart disease, but it increases the risk of breast cancer. As we go through, as we develop and go through menopause, our estrogen levels drop. And then we have these other things like our risk of type 2 diabetes would go up, for example. It can prevent breast cancer in older women and things like that. So it's a fine line. So in terms of fibroids, it made sense to begin looking at the whole aspect of hormone biosynthesis, how these hormones form. One of the things I remember quite distinctly as an undergraduate student in Mona was learning about the pathway, and I showed you that earlier, for the synthesis of these hormones. a single base pair change is responsible for the switch early on between estrogen and progesterone. Okay, and so these things are intimately linked. So we began looking at the genes associated with estrogen biosynthesis, the making of the hormone estrogen. And our methods are pretty simple. We sample some blood, we extract DNA from that blood, we do PCR, and I'm sure many of you now know what PCR is. We've gone through the COVID-19 pandemic situation in 2020.
Yeah.
We take an enzyme, and then we restrict that, we cut that up into small pieces, and we compare the profiles to our genes and do some data analysis. Okay, so that's what we do. And our first paper in 2014 led us to say that, yes, there's no significant difference with this particular gene. Let us continue looking at others.
Right.
And so with the help of another postgrad student, a year later, we began looking at CYP1B1, the one that's later down in the pathway. And we found again that this was pretty interesting, that we didn't find any significant linkages with the CYP1B1 and that variant. However, among the cases, there were more people with that variant. And the question was why?
Why, yeah.
And so when we began to think about it, we thought with many of these studies, you need very, very large populations, a large number, to make them significantly meaningful in the population, because we're talking about population, right? But Barbados is a small population. It's difficult to get people enrolled in these studies. And so our numbers were, we had about 150 persons, I think, around there in this study, in this particular study. And so we couldn't make any great, right, but statements about that, but we could see trends. And what we saw was that there were more people who had fibroids who had the CYP1B1 variant, a particular change in one of the amino acids in that variant. And so we continued our studies, and we began to think about how does this compare with the earlier work done in 2012 that showed that white women have this CCD57 or this fatty acid synthase link and UL. And again, we repeated that study in our population and showed that there was no relationship between that CCD57 or the gene responsible for fatty acids that is making up lipids and uterine fibroids. However, this was also shown by other persons who also, since our study, had worked with the same gene and the variant in Afro-American women. Again, numbers were small, but they showed something similar. So we felt fairly confident when we got our result that for Black women, this link was not there. We continued to do the work, and we decided to, at this time, the techniques improved, and we began to look not at a single variant, but grouping the variants together to form a profile. So for example, this is called a haplotype, when you try to find multiple variants. And so we built haplotypes for CYP1B1 and CCD57, because if you're looking for a single gene, it may not be there, but maybe if you group the many of these changes together, you may see something. What we did find was that, yes, again, the cases or the persons with fibroids had more of the haplotype profile called a GTA haplotype, and I'm going to show you this here. The GTA haplotype was quite prevalent among our cases. The high frequency, you can see about 72% of the population had it, yes? And although it was not associated with the risk, a high risk of fibroids, when we looked at the data again, it was protective.
Well, this has all been very, very fascinating. For those who might not be versed within the scientific field, how can we make this more accessible to others? What would you want them to take away from this research at the end of the day?
I understand. And so as a scientist, you know, we tend to get bogged down in the minutiae sometimes, what we're doing. So the big picture is that we now have an understanding of the incidence. For example, before, when I began the study, we didn't have a clear understanding of the incidence. It wasn't being measured in Barbados. We now know something about the incidence. We now know something about genes that may or may not be associated with our population in Barbados, and we can take that into Afro-Caribbean people living in the region. We now know that, and we've submitted this data to the databases, for example, what is the molecular profile, for example, of some women who have fibroids within our region. So while we cannot treat the disease, we can explain a bit more about people within our region, how they're at a molecular level, cellular level, what their profile is. And that's important for the clinician in understanding possibly how to treat.
Exactly. Right. So it's not doing the direct treatment, but it's supporting the pathway to treatment, for lack of a better term.
Yes, because one of the things that bothered me, and again, got me on this road to this research as well, was that when I began to ask about what the options were to alleviate, I was told hysterectomy, and that is the treatment. For a young person, that may be too aggressive, and I feel so. Some doctors feel so, some don't.
Yeah.
But in our population, in the Caribbean, that is the first treatment. There are options. There are so many symptoms associated with fibroids.
Yeah.
And these symptoms, what I want people to understand, or what I want our local governments to understand, is that they cost you work days. It is costly. And if we think about the fact that so many of our women are in the workplace and have to take days off at a time because of the symptoms of this condition, while it may not kill you, it affects your functioning. And so there is a cost. And in the US, they measure this. We don't measure this in our population. If we can begin to understand the impact of this and how our women, how we can get to a pathway to treat them, not just use a blanket treatment that's been used for decades and so forth elsewhere, I think we will be able to, I don't want to say alleviate, but to ameliorate the situation that we have with so many of our women in the region suffering daily from this condition.
Thank you, Dr. Alleyne. You have basically, in your own way and in your words, highlighted one of the main points that the BAEP has been trying to iterate time and time again. Yes, there's nothing, mortality is not necessarily associated with these conditions, no, but the level of morbidity, the the absenteeism, presenteeism, impact on your social well-being, your productivity, it comes at a cost. And to your point about hysterectomy, there are also side implications with hysterectomy too. So
many implications, yes.
Yeah, so you need to be aware of all of the options that can be provided to the women in your population who are coming to you as a healthcare provider for these conditions that, again, we did not cause them to happen to us. They're just happening to us. Now we need assistance in managing them. So thank you so much. You've done a lot of research. Those were what, 3 or 4 papers that you did, and that's a really short space of time, by the way, to do all of that work. So thank you so much for doing that work, in Barbados and placing Barbados on the map as it pertains to this type of research. What has been your most, I guess, significant, surprising, important scientific finding in your opinion?
That is a very interesting question, and I must say my journey is not done, so I don't think I can answer that. I would say, it's difficult to say what's my most significant finding. I would think that it is that, for example, when we found out that the CYP1B1 and haplotype GTA was protective, that was a mind switch for us, because we never thought we would find, we were looking for a direct association with risk. Mm-hmm. But instead of finding that there was a risk, we found it was the opposite. The fact that it was so prevalent, it wasn't that it was a bad thing, it might be a good thing. Mm-hmm. So for this particular project, that was significant for us. And so that was a surprising and significant finding. But I really don't think of my work in those terms. So it's a difficult question to answer.
A lot of people do. I do. So thank you for that. I can say for sure with BAEP, a lot of the papers that we've written, we've referenced your studies. Yes, thank you
very much.
Because again, we want to be able to highlight the work that is being done in the region on our populations with these conditions: uterine fibroids, endometriosis, adenomyosis, polycystic ovarian syndrome, because again, whilst the research in North America and Europe is critical and is important, it might not necessarily be representative. So it would be remiss of us not to utilize the information that we have on our populations in the work that we're doing, which is, where your research comes in. So thank you so much. So how can interested persons access this research if they want to learn more about it?
Well, as indicated, I work at the University of the Western East at Cable. So if you go to our website, you will find some of these papers listed in my profile or in the departmental webpage. And I must admit, you have to access the journals where they're published. They're not freely available, some of them. And so that's where they are. But you can also contact me, and I can also send you copies as necessary. Okay. By email if you are interested.
That's appreciated. So thank you so much, Dr. Alleyne. This was fascinating. I love the fact that I was able to go back to my biochemistry roots. I've been away for a while. I won't lie. I've been away for a while, but I appreciate that you're able to show the importance of, sometimes we think science is so far-fetched, and there's a disconnect between science and the implications in the general society. So I was happy that you were able to show the reasoning and the relationship between the research that is done, and then how it could support clinical trials and treatments that will be applied to the general population. So thank you for that. Thank you
so much for giving me the opportunity. I really appreciate this, and I simply, honestly, see what I do as a minor aspect of a bigger story. And so thank you so much for the opportunity.
Thank you. It's not minor, it's just part of the puzzle, but it's definitely not minor. So thank you. This is our very last BAEP Talks for the season. And so for today, we're going to wish everyone happy independence. Have a great holiday season, and we will see you in 2021. Thank you so much. Bye.
Thank you, and happy independence to all. Bye.
Thanks.